Most welcome my friend in our channel. We are coming back again some informations about the toxins. What is the toxins, which type of toxins, types of toxins, what is the basic definition of the toxins, why the microorganisms cause the it is used against the animals or plants as a defense mechanisms. So we need to understand basic things. It is a type of poisons that can cause any harms to either plants, animals, origins, or they can be either derived from the microorganisms, because toxins may be chemicals, may be human made, may be synthetic, may be another types, but some type of very toxic chemicals produced by the microorganisms. So we are here to discuss these microbiological originated toxins and toxic chemicals that cause some effect on human body system, and these toxins act as a antigens because these are foreign origins. So when it come into contact with the any living system, human body system or plant body system, they can be act as antigen molecules. Here we are understand the microorganisms toxins produced by these bacterial systems, fungi systems, and these are the toxins that can be different types, either the cytolytic toxins, here the cytolytic toxins are there, and the enterotoxins are there, and we are here to understand different type of toxic shock syndromic toxins, exfoliative toxins. So we need to discuss one by one. Here we are only to discuss in this video cytotoxic toxins, and later we will in the next video, so we will describe briefly how they can affect our cell systems and what type of the effect in body system. Just like botulism toxins is the neurotoxins and most potent against the neurons or the human systems. They can cause the different type of disease in our body systems. So we will discuss about later one by one in each videos. Now we will go for understand cytotoxins. Cytolytic toxins are membrane active substance and cause 4 type of the hemolysis in our body systems. 4 type of the hemolysis in cause the human system, and one is the leucocidins. What is the leucocidin? We will discuss in the later sections. So we will start what is the hemolysins, which type of hemolysin in our body systems, which type of hemolysis caused by the bacteria. Generally we are understand, we know about the alpha-hemolysin, beta-hemolysins, or gamma, delta type of hemolysins we are generally noticed when we are going to understand bacterial microbiological analysis of these things in the lab. So hemolysis are the type are the chemicals which are composed of the lipid molecules and some proteins molecules that cause the lysis of the red blood cells. So these chemicals that cause the disruption of the cell membrane of the RBC. So the inside the chemicals present in the cytoplasm of RBC that come out outside after the disruption of this RBC molecules. Although this lytic activity of the microorganism derived from the lysis is the blood may be of the great important nutrient acquirement, because this lysis some bacteria need some nutrient inside the RBC. So these nutrient after disruptions they come in directly contact with the bacteria. Bacteria can use these type of things, but the lysis of the RBC there is a cause disturbance in the host systems. So host molecules are feelings, some things, infections, some type of the disruption in their blood circulation systems. However, hemolysis is often capable of lysis of RBC in vitro. We will also study in vitro technology. अगर हम इस चीज़ को स्टडी करना चाहते हैं तो हम in vitro भी इस तरीके की स्टडी कर सकते हैं जाकर. So we are here understand first things is the hemolysis. Alpha type of hemolysis क्या होता है? Alpha hemolysis is the lysis that most important among all of them, either beta or delta or gamma. The protein become inactivated in this around 70 degrees centigrade, but reactive 100 because of this 70 to 60 degrees centigrade toxin combination heat labile inhibitor and denature in these types. So in this whole all these things is that the alpha hemolysins here is telling that alpha toxins lysine, the activity become inactive in this protein at 70 degrees centigrade, but reactive paradoxically 100 degrees centigrade. But it is just the proteins are described here. The actual mode of action of this protein is inhibit, but the protein is temperature labile. According to temperature, the activity may be inhibit or may be controlled. This is the not very important factor. Another factor of this alpha hemolysis, just we can take an example of the bacteria Staphylococcus aureus secrete alpha hemolysis. This type of the lysins monomers, outer membrane of the RBC molecules be destroyed if there is a large number of these alpha hemolytic lysine molecules come together around these type of things, and they forms the membrane, cell membrane of the RBC molecules, permeables, so all chemical inside them come out, where all these chemicals, either water molecules, ions, some organic molecules, these organic molecules are used by the bacteria for their growth. Rapid discharge of these molecules such as ATP molecules also come out inside. So ionic gradient and irreversible osmotic potential develop. They can cause the rupture of the RBC molecule or host cells molecule. So if there is this type of cell is there and there is all the molecule come out outside, and so all these things the cells become ruptures. One way hemolysis is erythrocyte is forming pores या phospholipid layer. This is ये एक प्रकार का ऐसा एग्जाम्पल्स दिया गया यहाँ पर कि आफ्टर द hemolysis all the chemicals, चाहे RBC molecule जो भी सेल्स होगी उसके रेप्चर से जितने भी इनसाइड केमिकल्स होंगे वो सारे बाहर आ जाएंगे, और बाहर आने से जो ब्लड वेसल्स है they will face the problem of the another potential development. This pore forming, because there is a large number of the lysine molecule around the cells, so major mechanism of this toxin cause the damage of the cells. So the number of cells are damaged. So as compared to the number of living cells, the RBC, the number combination become disturbed. So this disturbance cause the illness, yeah, something fever like development inside the human body systems. It is expected now that here we are coming to understand sufficient doses alpha toxins in the mammalian cells. It is also just like this. Here we are not going to details. We will understand which type of this. If you can see this diagram, this chemical cycles, if there is disturbance happen, all the molecules come outside, and these calcium molecule, this is the alpha toxins, and all these another potassium channel disturbance, everything is disturbance. So you can see these pictures where you can see there is a clean in the blood agar plate. The bacterial growth is causing the disturbance and the killing of the all blood vessel cells, or if there is a present in this blood agar molecules, they can utilize the cells in everything. Now we are understand second beta hemolysis. What is the beta hemolysis? Beta hemolysis is the phospholipase toxins, where there is a lipid and proteins molecules, but here is the phospholipase, that is a phosphate molecule and lipid molecules. They can combine and form the toxins secreted by the also SRS molecules, SRS bacteria, and we can study in the sieve erythrocyte, where we can go for the lab and understand the basic principle behind this. How can this toxic mechanism is discovered by the hemolysis sphingomyelin, which is account for the 50% of cell membrane. You know that in the body cells RBC molecules are present. This chemical is around 50%, and this beta hemolysis specifically act on these chemical molecules. So the phosphoryl choline due to release of the organic phosphate from this, because this is a phospholipase. So if there is a phosphate molecules, they can cause the disturbance and release these phosphate molecules, and sphingomyelitis become dead, the cells become ruptured. So you can see the picture here where you can show this type of the structure inside Petri plate. You can find, you can observe, this is the beta hemolysis molecule, beta hemolysis. So it is a different from the alpha hemolysis and beta hemolysis. Clear in case of the alpha, here is not clear, here is just like clumping. So you can see. Another is the gamma hemolytic process. Gamma hemolysis is composed of two separate proteins. Here only the proteins molecules are presented. Only the proteins are presented. Both protein necessary for hemolytics. First the gamma, alpha and beta. These are the lipid molecules, but here only the protein molecules. Gamma hemolysis only the forming of the pore-forming toxins. They can as like to the similar alpha hemolysis, just like similar, but it is unique bicovalent toxins compared to the beta hemolysis. So you can see the picture, you can find something very clustered like structures as compared to the alpha and beta. There you can identify these things. In higher affinity phosphocholeins with short saturated acyl chains, especially they can colonical forms, and colonical form of this type of hindrance activities are occurs. So you can differentiate other molecules by using this type of shape and structures when you are going lab analysis. Alpha, beta and gamma, you can see the pictures and you can identify which type of the hemolysis is going on this type of the Petri plate. Now you are coming the delta hemolysis, is the detergent like activity, and membranes are just like detergents. They can kill all these things, denature all the molecules. Erythrocyte, leukocyte, macrophage, Petri plate, all the cells can be detergent like, just like detergents. They can just denatures the proteins molecule, just like other detergent activities. In case of, you can study in the organic chemistry how the detergents activities happen. So you can do in Google's, you can find out. Last is the leucocidins. Leucocidin is also called as the pentone valentine toxins after discovery, because these are scientist names. So the two component toxins are this gamma lysins, like just like gamma lysins, two component S and E are presented. Such bicovalent membrane toxins are staphyloleucocidins, gamma lysins, and combine this type of this is the very rarely found in the some bacteria like a staphylococcus, some other bacteria, leucocidins, and the two protein molecules are present. They are toxic protein molecule S and E, and they can forms the combinational and they can attack on them. And this type of structure you can see, grape-like structure is, you know that staphylococcus is forming. So such type of the dead colonies are presented, and in this whole mechanism you can see the beta barrel protein molecules are denatured by this leucocidin molecules. So this is the whole type of the first step of how the such type of hemolysis present in the bacterial cells, how they cause the host pathogen detergents. This is the one of the step we will discuss here, the cytolytic toxins, and later section we will go for the enterotoxins, toxic shock syndrome toxins, and exofolate toxin molecules. So overall these things, how the bacteria cause the toxins release, and which type of toxins are present inside the pathogen molecules that can be, you know that toxins are secondary product, metabolitic product of the bacteria. They can be many types. So we are here to read and understand all these things. Thank you so much for giving your time to and watch me. If you really like, please watch and subscribe my channel.